Innovations in precision medicine are transforming the paradigm of cancer care. Predictive biomarkers enable identification of patients who are most likely to derive therapeutic benefit from a particular treatment, such as PD-1 immune checkpoint inhibitors, and form the basis of precision medicine. Cancers are remarkably diverse. Tumors originate from different types of tissue and may have distinct differences in anatomopathological and genetic features. There are also key differences in the tumor microenvironment. Some tumors contain high numbers of tumor-associated immune cells, such as lymphocytes and macrophages, whereas others contain very few tumor-associated immune cells. Pembrolizumab has a broad clinical program. PD-L1 expression is used in some tumor types, where appropriate, as a predictive biomarker to identify patients who are most likely to derive therapeutic benefit from pembrolizumab. In non-small cell lung cancer, the evaluation of PD-L1 expression on tumor cells has been shown to be sufficient to predict benefit from PD-1 inhibitors, such as pembrolizumab. In other cancers, PD-L1 expression on tumor cells alone is insufficient to predict benefit. The inclusion of PD-L1 expression on tumor-associated immune cells is required to predict benefit from PD-1 inhibitors. Currently, there are two different PD-L1 scoring algorithms for pembrolizumab to account for the difference in tumor biology. These are the tumor proportion score, or TPS, and the combined positive score, or CPS. TPS is calculated by dividing the number of tumor cells showing partial or complete PD-L1 membrane staining by the total number of all viable tumor cells present in the sample times 100 to give a percentage score. The key difference between CPS and TPS is the inclusion of PD-L1 positive tumor-associated immune cells in CPS scoring. CPS is calculated by dividing the number of PD-L1 positive tumor cells and tumor-associated immune cells by the total number of all viable tumor cells present in the sample times 100. The types of immune cells included in the algorithm are lymphocytes, including T cells and B cells and macrophages. These immune cells are directly associated with response against the tumor. To determine TPS, the first step is to distinguish tumor cells from tumor-associated immune cells, as both may exhibit PD-L1 positive staining. Score only viable tumor cells. Partial or complete membrane staining are considered PD-L1 positive. Exclude all other cells from scoring. These include tumor-associated immune cells, normal cells, necrotic cells, and debris. Also exclude tumor cells with only cytoplasmic staining. Calculate TPS by determining the percentage of PD-L1 positive tumor cells relative to all viable tumor cells present in the sample. To determine CPS, the first step is to identify viable PD-L1 positive tumor cells and appropriate PD-L1 positive immune cells for inclusion. Various immune cells may exhibit PD-L1 staining, but only tumor-associated lymphocytes and macrophages should be scored. These are located within the tumor nests and adjacent supporting stroma. Tumor cells with partial or complete membrane staining are considered PD-L1 positive. Tumor-associated immune cells with membrane and/or cytoplasmic staining are considered PD-L1 positive. The CPS is calculated by dividing the number of PD-L1 staining tumor and immune cells by the total number of viable tumor cells times 100. CPS is expressed as a numeric value and not as a percentage. Tumor diversity dictates the need for tumor-specific scoring algorithms to predict which patients are most likely to derive therapeutic benefit from PD-1 inhibitors, such as pembrolizumab. MSD is committed to innovation in precision medicine to transform the paradigm of cancer care.